first off, proteins and their function. Proteins are like humans. There's all different kinds, all different shapes, all different sizes, okay? So unlike, again, functional groups here, we are now not talking about a piece of a molecule. We're talking about gigantic molecules. Proteins can be short peptides. They can be huge. They can be thousands of amino acids long. They can be a few amino acids long, all different kinds. So don't box it in and think, oh, you got to have this to be a protein. No, they're widely different, but there are a few things that kind of hold this class of biological molecules together. And it's with what they do and what they provide to our body. So your body has, anybody know roughly how many proteins are within the human body? It's a big number. Anybody want to guess? I actually really want somebody to guess. I want to know what y'all think. How many proteins are in our body? Anybody? The price is right. Y'all are quiet this morning. Maggie has a guess. Robin has a guess.
No, I don't. I'm exhausted. Hold on.
1 million?
Thank you, Maggie. At least we got a number out. Okay.
A trillion.
Do what?
A trillion. A billion.
Not that many. Let's calm down, everyone. Okay, 60,000. All right. But still, that's a lot of proteins, right? So there's roughly 60,000 proteins in the human body, give or take. But what do? Okay, so they provide structure and support. They help hold us together, our bones, our organs, our tissues. They regulate like hell. They regulate everything we do, okay? It's like the parent you never wanted. They literally are in charge of everything in our bodies. They're the workforce of your body, okay? So they control the process of metabolism. They act as hormones and enzymes. They're constantly regulating when we do what we do and how we do it. As far as it comes to, I mean, any system you can think of, digestive, circulatory, your nerve systems, all the stuff. Storage and transport, okay? So they help our body store things. They help our body release things that are stored. They help transport drugs and molecules from places they need to go, signals. And then protection within our immune system. Okay, so you can thank your proteins when you go to bed tonight for all they did for you today. But here's two pictures, obviously of just two individual proteins, but you can see how structurally different they are. Okay, what is the top one? Keratin. What does that look like to you guys? What can you see in the structure for keratin?
Like coils?
Uh-huh. We call these helical strands, okay? You'll hear me say the word helices, okay? So these are called alpha helices. So you have some proteins that are just helix in structure, okay? And they're just coils of helices. Keratin is one of those. And immunoglobulin G, okay? So keratin, by the way, is a protein in our hair. It's support and structure for hair. Immunoglobulin G, this is part of our immune system. And you can see the structure here looks a little more globular and not as like, what's the word I'm looking for? Kind of fibrous, the way keratin is. But anyways, I just want to give you two examples there. This is a table. You need to essentially know these types and kinds. Okay, so if I ask you, what does a contractile protein do on the exam? You would need to be able to just give me a short, statement about what they do. So know these kinds and types of proteins. We've got enzymes, hormones, storage proteins, transport, structural, protective, contractile, okay? You can see the function of each of these. Enzymes catalyze biochemical reactions. And by the way, we're spending the whole day on enzymes tomorrow. Okay, that's chapter 19. But enzymes, they're proteins, okay? It's just their function that makes them unique. So when we say catalyze, they're not a reactant in the reaction that they're catalyzing. They're just kind of a helper, and they're not changed. They're not part of it. They're just helping the reaction happen. Hormones, we are very familiar when, as what hormones do in our body. We have all kinds of hormones, but they're regulatory proteins again. Okay, so You know, how does your body know when to stop growing? Have you ever thought about that? Like you grow. I have a 1-year-old and a 2-year-old home, and they're growing out of outfits left and right. They're growing all the time. But eventually our bodies stop growing. Why? Okay, why don't we just continuously grow our whole lives and become 60 feet tall? That would be cool, right? Well, we don't. And so that's because we have hormones that we have growth proteins that, you know, those passageways and those things in our bodies are turned off at a certain age. And that's just one example, but that's like what hormones do. Okay. Why at a certain age do guys, their voice kind of lowers, and girls develop different features. Okay. That's all to do with hormones. And so these turn on and off as our bodies need them. But these are proteins, storage proteins. Okay. Make essential substances available when we're needed. I'm not just going to read this word for word. You can do that, but you need to know the functions of these classes of proteins. There is examples here. I don't necessarily need you to memorize all these examples, but I think it is helpful to have an example there. Okay. But I do definitely, I'm going to put a star once you know the function of these. Okay. So based on what we just talked about, I've got a poll question. You tell me what you think based on the little description for alcohol dehydrogenase and then oxytocin and the other one. What kind of proteins are these? So alcohol dehydrogenase found in liver cells converts ethanol into acetaldehyde. And we talked about alcohol dehydrogenase when we introduced alcohols. We talked about fetal alcohol syndrome. And we looked at this exact reaction. It's an oxidation reaction. So what kind of protein is alcohol dehydrogenase? You think it's an enzyme, hormone, storage protein, transporter, protected protein? Read back through the functions if you're just not sure. And then cortisol and oxytocin. Cortisol level rise under stressful conditions, and oxytocin can induce relaxation and romantic feelings. What type of proteins are cortisol and oxytocin? Just give me your best guess. Do you have no clue? We will talk through it. Okay, one more. Whoever's last, get your vote in for us. I'm just gonna, okay. And Paul share results. All right. So these are the results for number one. Most people said an enzyme, but there were some other choices selected as well. So let's think about this. Alcohol dehydrogenase, it converts, that's kind of the key word there, ethanol to acetaldehyde. So all this enzyme is doing is making this reaction happen. Okay. That's the purpose of its whole life. All right. Is to just convert ethanol to acetaldehyde. So it catalyzes, this would best be classified as catalyzing a biochemical reaction. Okay. So alcohol dehydrogenase is an enzyme. Okay. It's not transporting ethanol throughout the body. It is not storing ethanol. It is not making our body do any mechanical work. It is simply metabolizing or breaking down this molecule. All right. It's not making ethanol available. It is just catalyzing this conversion of ethanol to acetaldehyde. So that is why it's an enzyme. And then cortisol and oxytocin, these have to do with releasing or signaling to our body when we are under stress. Okay, then this is released. Okay, certain feelings can release things in our body, and so sorry, I'm trying to move my poll window. Most people said hormones. Okay, one person said storage proteins, but that is exactly right. These are hormones. Okay, they regulate body function. They send messages, and messages often are tied to our emotions and how we feel when our body is messaging or signaling certain things to our brain and throughout the body. Okay, so these are for sure hormones. Any questions on that? Okay. I wanted to show you this table. This is stuff you already know, but we've talked about a lot, and I kind of want you to hone in if you're wondering like, okay, what, I need to stop sharing the results one. What from all this past stuff do I really need to know moving forward? All of it in pieces, but really essentially when it comes to functional groups, these are the ones you're going to see most often from now on. Okay, so the ammonium ion or the amino group. Okay, those are derivatives of amines. Okay, nitrogen. The hydroxyl group, carbonyl group. You hear me say the carbonyl carbon a whole lot. You're going to keep hearing me say that. Carboxyl group and carboxylate anion. So that was on the exam yesterday as far as knowing that these carboxyl groups are acidic, and when they lose their hydrogen, you are left with the carboxylate anion. Amide group, we're going to see that a whole lot more. Protein amine bonds are amide bonds, so you're going to see those. Carboxylate acid esters. Okay, phosphates. We really haven't talked about phosphates a whole lot. I mentioned them at the end of carboxylic acids, esters, and amides, chapter 17. But what did I do? Okay, I'm going to zoom in. So the phosphate group is this PO4 group. Okay, I just want to remind you, and it is bonded to a carbon. And these phosphate groups can be joined up by what we call phosphoester bonds. Okay, you could think of them like ester bonds, but instead of a carbon to oxygen to carbon, it is a phosphorus to oxygen to phosphorus. Okay, so we're just linking up these phosphate groups, and we would call this well, that's why you see the prefixes here, mono, di, and tri, is just indicating to you the way mono, di, and tri would indicate like the number of methyl groups that are substituents. Here it's the number of phosphate groups that are substituents. So a diphosphate, triphosphate is probably what you've heard with ATP, adenosine triphosphate. Okay, so I just want you to be familiar with that. We're going to talk about phosphorylation reactions, especially tomorrow with enzymes. And that is just the transfer of, I just want you to know this group, a phosphate group. Okay, and there's negative charges associated with 2 of these oxygens in a particular phosphate group. So it often carries a -2 charge. Okay. Hemiacetal groups. Okay. Acetyl groups, and then thiols, disulfides, or sulfides and disulfides. Okay. All right. Just want to show you that. Now we're really going to get into proteins, or sorry, the building blocks of proteins. So you guys, everybody's heard of amino acids before? Probably. So amino acids are, simply put, the building blocks of proteins. Now here is the base skeletal kind of nucleus of an amino acid. And what do you see? You guys tell me, what do you see? What groups do you see? Somebody just name one group that you see, one functional group.
An amino group.
Yeah, right here. Thank you, Robin.
And then there's a.
Nope, Robin can't do the next one. Somebody
Okay.
else.
Carboxyl.
So, somebody said it.
Carboxyl, is that what you were talking about?
Is that Maggie? Who said that?
Abby.
Oh, Abby, sorry. Okay, yeah, carboxylic acid right here. Okay, so here's what you need to know. Every amino acid has an amine group and a carboxylic acid, and they're oriented just like this. The carbon in between is what we call the alpha carbon. Every amino acid has an amine group, an amino acid, or an amine group, an alpha carbon, and a carboxylic acid, and they're in this order. Okay. We typically draw them with the amine group on the left, the carboxylic acid group on the right, and the alpha carbon in the middle. Okay, that alpha carbon has a hydrogen branching off. Every alpha carbon has one hydrogen, but where amino acids are different is this little R right here. I'm just going to highlight that, the R. This is what we call the side chain. These can be wildly different. Okay, we have 20 amino acids that we know of. And we have 20 different side chains. Okay, Cameron, is your mic still not working? If you could join us with your phone so we could have your mic, that would be awesome. Sometimes I don't see your comments and I feel bad. I guess your mic's still not working because I can't hear you right now. Cameron, you there? Okay.
Yeah, it's not working.
Yeah, I saw his comment. Try to join us with your phone next time because it was working that one time with your phone. All right, so 20 amino acids, unique side chains. Here's what you're gonna need. You have to memorize all 20 amino acids. Yep, you do. They're very, very important. Why? Because they are. It's one of those things. So here's what I'm gonna help you do. I would make flashcards if you need to, but if I have to ask you to draw out the structure of any one of these 20, you need to be able to do that. So here's what we got. These are non-polar amino acids with neutral side chains, meaning they don't carry a charge, okay, or a functional group that could be charged. You do see, again, this part of every amino acid is the same. The amine group, the alpha carbon, the carboxylic acid, and at physiological pH. So all of these amino acids are being shown to you at pH 7.4. Okay, so this is what they would look like in the body. Okay, the amine group, what do you notice about it? It's always charged. It's an NH3+, it's an ammonium ion. Okay, you see the positive charge on all these amine groups of every amino acid. The alpha carbon is just normal. The carboxylic acid though, what do you notice for this? On every amino acid, it has given away its hydrogen. It's in the carboxylate anion form. Okay, so every amino acid, the backbone, if it's by itself, is charged. If it's bonded up in a peptide, it's a little bit different. We'll get to peptides in a minute. But individual amino acids, this is how they look. So what it shows you just underneath, so if I zoom in on alanine, the part in green, I'll highlight it in green. The part in green, this is the side chain. And this is the part that you can tell is different for every amino acid. So alanine just has a methyl group, okay? Glycine is even smaller. It's got just a hydrogen. Isoleucine has a 4-carbon group. Very similar to leucine, just the thing that's different is the placement of this methyl group on that extended carbon chain, okay? But all non-polar, just carbons and hydrogens. Methionine. What kind of functional group do y'all see on the side chain of methionine?
There's a thiol. No. Is that
Close,
thiol?
Abby. Keep go one more down the list. So it's either a thiol, a sulfide, or a disulfide.
Sulfide?
Sulfide, yeah. A thiol, remember, is SH, and these are terminal, okay? But this sulfur is tucked in the middle, so that's a sulfide group, okay? So methionine will be able to do some things that other ones can't. In addition to, I think I have it down here, is cysteine over here? No, cysteine's on the next table down. Cysteine is another one that has sulfur, okay? Then you got phenylalanine, which is a CH2 with a big aromatic ring. So that's quite a large. I want you to notice the size difference on the side chains. So phenylalanine is kind of thought of as a bulky, large side chain. Very hydrophobic, very nonpolar. Proline is very unique. Proline actually is a ring structure that connects, I mean, y'all can see it connects the alpha carbon and the nitrogen on the amine group. So anytime proline is within the structure of a protein or a peptide chain, it causes it to kink, okay? Because it's a little bit turned. It's not just flat, right? So it causes protein structure, it causes kinks, and it allows proteins to coil. You'll find them in alpha helices a lot because it helps them coil in alpha helix. So it affects structure, causes to turn or kink. All right. Specifically, if we're looking at primary protein structure, just the sequence of amino acids. Tryptophan. Tryptophan is also another large nonpolar amino acid. So you've got the Ch2 group, but then you have this heterocyclic structure with the nitrogen in it. It's an indole ring. If you remember from amines, we have imidazole is the ring structure for histamine, and this is an indole ring on tryptophan. You don't have to memorize the name, but that's what it is. So you just see this aromatic ring, and you see this other ring structure with the nitrogen group right here branched off of it. And valine, okay, the way I remember valine is I tend to write it like this. So here's my alpha carbon, and then here's the CH and the CH3 and the CH3. I remember valine like this. It makes a V, an upside down V. Okay, the side chains do. I'm always looking for shortcuts on how to memorize things. Okay, in addition to just the name, by the way, you see the 3-letter shorthand notation. So every amino acid, you'll see this when you do practice problems in your textbook. Every amino acid has a 3-letter short, 3-letter notation. Usually it's like the first 3 letters of the word, but sometimes it's not. Okay, so valine, just VAL, that makes sense. And then a V for a one, it also has a 1-letter abbreviation. Usually the first letter of the word, but some amino acids start with the same letter. So their 1-letter notation isn't always the same letter. The first letter of the word. Tryptophan is a great example here. Its 3-letter abbreviation is not TRY, it's TRP. And then it's a single letter notation of W. The way I remember tryptophan is the base of these rings make a W, if you kind of outline those. Okay. So these are all nonpolar. And by the way, we need to fill in these blanks over here. Nonpolar side chain functional groups are hydrophobic. And they will interact with London dispersion forces. Okay. So when they're around each other in a protein structure, when it's all folded up, if there are nonpolar amino acids next to each other, they'll interact. They'll add structure and stability to the protein by interacting with London dispersion forces. And usually these amino acids are tucked on the inside of a folded protein. Okay, but we'll get to that later. Okay, next group, polar side chains, but also neutral. Okay, so these are polar, meaning they have some electronegative atoms that you did not see in the previous part of the on the previous slide, but they're not charged. Okay, you're not gonna see plus and minus charges. You just see the oxygens and the sulfurs and the nitrogen. So for example, asparagine, 3-letter abbreviation ASN. What do you see? You have, what is this group right here? Somebody can tell me what that is. It looks upside down, but nonetheless, it's what kind of functional group?
Is it an amino?
It has an NH2, but that NH2 is an amino, Robin, but it's bonded to a carbonyl carbon. So what do we call it when there's a nitrogen bonded to a carbonyl carbon?
Oh, an amide.
Close. Change one letter. Change one letter. Somebody help her out.
Amine?
Did you say the same thing? It's not an amine.
No, I said amide.
Oh.
AMIDE.
Yeah. Amide is how I say it. Sorry.
Oh, I'm sorry.
Yes. No, you're good. Yeah, that's an amide. Okay. Cysteine. Y'all see the thiol group on cysteine. All right. So remember, when you react to thiol groups, you can form a disulfide, and we're going to see disulfide bonds when we talk about protein structure, like the different layers, a few slides down. Okay, glutamine, another amide bond or amide functional group on glutamine. It's just a little bit longer than asparagine. Asparagine and glutamine to me are very similar. The only thing different is the additional CH2 group on glutamine. Okay, before you get to the amide functional group. But you got these electronegative atoms, so it's just polar but not charged. Serine, question?
Yeah, are we gonna need to know the functional groups? Because like I just don't want to write them down if we don't need to know them in it, because that'll just confuse me. So, like, will I need to know that that's an amide in the asparagine or whatever the first one we did?
That's an amide over here? You already know that, right? I mean, we spent a whole chapter talking about amides and
Yeah, but will like we need to like label it?
on it. Oh, I mean, I honestly have not even written the exam yet, so I don't know what's going to be on it yet.
Okay.
But that, I mean, that shouldn't, I wouldn't think that would give anybody any like headaches or hiccups if I ask you what functional group is on the side chain of asparagine. If you know the structure of it. Well, so the important thing is that you're able to draw these side chains out if you need to. Okay, serine just is an alcohol group there, CH2OH. Threonine, also very similar to serine. Okay, it's almost, it's just got this additional methyl group branched off, but it's got the alcohol OH group as well. And I'm trying to point these out, Robin, to just help you guys see how certain ones are similar. In the long, I know this is a lot of information right now, but in the long run, that will help simplify things if you can group amino acids and remember, oh, there's two that have amides or there's two that have alcohol functional groups. Tyrosine is another one, so 3. But it just has that aromatic ring before the alcohol group. Okay. These blanks over here, so we know that polar side chains are hydrophilic. And will interact several ways, okay? So they will have what we call salt bridges. These are like ionic charges, okay? I'm gonna write that next to it. These are like ionic. Okay, they'll interact with ionic charges. That's really mostly the amino acids down here that we haven't named just yet. But then they'll have disulfide bonds. And hydrogen bonds. Okay, they can, many of these amino acids and their side chains can hydrogen bond. I also want to point this out. You will hear people say this if you're around biochemists or biochemistry very much. Structure determines function, and maybe you've heard that in like an anatomy and physiology class too, probably. Structure determines function. So the fact that all these different amino acids have a different structure will allow different functionalities or do different things in a protein, okay? Not just with its shape, but with how that protein then acts physiologically in the body. Okay, last group here is the acidic side chains, okay? So we now are gonna have these often will be charged in the body, okay? So at physiological pH, you see, look, all these charges. Now we have this, it was a carboxylic acid, but it's now just this carboxylate anion, okay? Aspartic acid, glutamic acid, the reason the word acid is in the actual name of the amino acid is because it has that carboxylic acid functional group. But look just underneath, probably what you'll hear me say the most is glutamate and aspartate. Why am I putting this ATE ending? Well, remember when a carboxylic acid loses its acidic hydrogen, we name it as a carboxylate 8 anion. Okay, so instead of our aspartic acid, aspartate is just the negative charged form of that. Glutamate is when it's negatively charged on that functional group. Okay, it's lost the acidic hydrogen. And then arginine, so arginine and lysine and histidine too, they all have positive charges. And histidine can also carry a positive charge as well, okay, if it receives another hydrogen, and it can. These nitrogens, remember, they have lone pairs, they can act as bases and receive hydrogens. But at physiological pH, histidine is, the side chain is technically neutral. So Okay. So 20 amino acids, 20 side chains. Please know the structure and how to draw out these side chains. All right, we're gonna do a practice problem. So I'm gonna skip over this practice problem. And just alpha amino acids are not important right now. But I have another poll question for these two. So now that we've talked about all the amino acids, or I've introduced them at least, which ones did we say, if you are just looking at the names, you might have to go back and look at the structures. Which amino acid is hydrophilic? And then the second one, which one is hydrophobic? So we've got isoleucine, phenylalanine, aspartic acid. So which one would be hydrophilic? Think about which one would be polar or charged, which are nonpolar. And then on the bottom, which one's hydrophobic, which one's water-fearing? So if water is a polar molecule, you're looking for the amino acid there that's non-polar. I'm still waiting on a couple. Last one, get your guess in. Okay, here we go. So most everybody said aspartic acid would be hydrophilic. So remember isoleucine, the side chain here. So we have the alpha carbon right here, alpha carbon, and then the side chain is a bunch of hydrocarbons. Okay, it's got like a long hydrocarbon chain, CH3, that might be C, and then CH2, CH3. Okay, that's isoleucine. So it's all hydrocarbons. So this is nonpolar. So not what we're looking for, not hydrophilic. Phenylalanine, also very nonpolar. It's got the big aromatic ring. So aspartic acid here, which is right on the slide above, what most people picked. Aspartic acid is charged, it's very polar. See those electronegative oxygens? So that one's hydrophilic. Phenylalanine was 2 slides above up here. This is phenylalanine. This side chain is totally nonpolar, which is why it's grouped up here with the we said it was a nonpolar amino acid. Okay. Hydrophobic, then, you are now looking for which amino acid is nonpolar. So glutamic acid and arginine are down here. These have acidic side chains. So glutamic acid and arginine are not hydrophobic. They are very hydrophilic. Tryptophan is the one here. That's what most people picked. Tryptophan is up here with the nonpolar group, okay? So that one is hydrophobic. All right. Good deal. Any questions on those? Okay, so those are the amino acids. Last thing you need to know about amino acids is chirality. So 19 of the 20 amino acids are chiral. Okay, if we're looking, this is a little bit of a different orientation of the side chain here. Here's the amino group. Here's the carboxylic acid or carboxylate group. Here, carbon 2 is your alpha carbon. So again, we normally write it with the amine group on the left, the carboxylic acid on the right. They've just oriented the carboxylic acid upward in this picture. But the alpha carbon has a hydrogen attached, and then every amino acid has this would be the R group, okay? And every R group is different, okay? So all 19 of the 20, the one that's not chiral, so the alpha carbon, by the way, that's the carbon that's chiral. That's what makes the amino acid chiral. You can get left-handed or right-handed, depending on what's different about these. What's different about the right-handed and the left-handed version? If you're looking closely over here, I'll highlight. So the amine group is on the pointing to the left, but here the amine group is switched. It's been turned around, pointing on the right. So it's a mirror image. And you cannot overlay these, they're not superimposable. And that's where we get the L and the D annotations, okay? So remember when we talked about monosaccharides, we were identifying chiral carbons in the monosaccharides, and we could get mirror images, and we have the D and the L forms, and we said that the dominantly used form in nature when it comes to monosaccharides is the D form, but here with amino acids, it's the L form. Okay, nature selectively uses the L form, right? Not the D amino acids. The one that's not chiral is glycine. Okay, notice for glycine, the R side chain is another hydrogen. So this alpha carbon, this is our alpha carbon again, has 2 hydrogens attached. So it does not have 4 unique groups. 2 of the groups that are attached to the alpha carbon are identical. They're both hydrogens, okay? The amine group and the carboxylate group are unique, but because it has 2 identical hydrogens, you don't have a chiral molecule, and the mirror images are superimposable. Okay. Questions on that? That's really all I'm going to say when it comes to chirality and amino acids. Okay. Acid-base properties of amino acids. So we have color-coded here, blue and red, the 2 acidic groups. So the carboxylic acid group, it can lose a hydrogen atom. So the one that can be given away is this hydrogen right here that I'm underlining. That can be given away. The amino group is basic. It can gain a hydrogen atom. So we can add a hydrogen atom to that nitrogen right here. When that happens, which is what you saw right up here, if I scroll back to the amino acids, this is what they look like, right? It had gained a hydrogen, it had lost a hydrogen. And when you have an amino acid like this, we call it, go back down, we have a special name for it. It's dipolar. It's a dipolar ion. It's got a plus. Dipolar just means it's got 2 different charges. And these are known as Zwitter ions. It's a weird word, Zwitter ions. They're dipolar. They have a plus charge somewhere in the molecule and a negative charge somewhere else in the molecule. Zwitter ions. They have properties similar to ionic salts. You can crystallize proteins. A lot of people study the structure of proteins by crystallizing it and looking at the crystal structure. Okay, so they can form crystals. They have high melting points and boiling points, and they're soluble in water. Very polar. All the charges are definitely reminiscent of an ionic compound, which we know ionic salts are soluble in water. Okay, so know those properties. Continuing though with this thought, let me remind you one thing about pH, okay? Because we're about to talk about how pH affects amino acids greatly and proteins greatly. So remember, when we talked in chapter 10, acidic environments have a high concentration of H+. That's what makes acidic pHs, if we're talking about the pH scale, we have a pH of 1, 7 is neutral, 14 is basic. So any pH between 1 and 7, this would be acidic. And this has a high concentration of H+. That is what makes an environment acidic, and that's why we give it an acidic pH. On the flip side, let me go over to different color. Basic environments have a high concentration of OH-. So any pH above 7 to 14, technically say this is basic is what we call it, and has a high concentration of OH- or a low concentration of H+. So just want to remind you about that, okay? Keep that in mind as we talk about pH. So how does that affect amino acids? Well, look at this figure over here. I'm going to keep this other side visible. Here's an amino acid, and it's a Zwitter ion. You see the plus charge on the amine group, the minus charge on the carboxylate group. In acidic solutions, Zwitter ions accept hydrogen. How do they accept hydrogen? Onto the carboxylate. So we see the plus charge on the hydrogen and the negative charge on the oxygen. So that carboxylate group will just receive that hydrogen back onto it. It'll be protonated. So now what kind of charge do we have? It's not a Zwitter ion anymore, but what kind of charge do we have on this amino acid? Just a +1 charge, right? Because of the amine group is still charged. In basic solutions that have a lot of OH minus, Zwitter ions lose their proton. Well, what proton are they going to lose? Well, they can't lose one from the carboxylate group. It's already lost its proton, but they can lose one from the amine group. And the amine group goes from a +1 and it becomes neutral over on the product side, okay? But overall, that's just the amine group that's neutral, not the whole amino acid. We still have overall, we have a -1 charge on the amino acid overall. I'm going to circle these. These are the overall charges. Because that carboxylate group still carries the minus charge. Okay. I'm going to keep going. Because of this property, we have something with amino acids called, I'm going to scroll down a little bit, called an isoelectric point. So we're about to talk about the isoelectric point, okay? So let me backtrack though and read these bullet points. The overall charge of an amino acid at any given time depends on two things. It depends on what side chains in the amino acid and the pH of the environment that the amino acid's in. Okay, I'm going to say that again. The overall charge of any amino acid at a given time depends on the particular amino acid present, what side chain it has, and the pH of the environment or the solution. And guys, this is really, really, really important. Tomorrow we're going to talk about enzymes and how enzymes catalyze reactions, and they have these really dynamic, amazing active sites that molecules bind to. And these active sites are completely dependent on this right here. Whether they have charged groups or non-polar groups or just polar stuff that's not charged, that all affects how the enzyme behaves and how it acts, okay? And we are highly dependent on enzymes. Some enzymes catalyze molecules. They can do like over 1 million molecules a second, okay? Doing the reaction 1 million times a second. And that's so important for our bodies. And it's based on this, what charge different amino acids will carry in the pH in our cells, in the pH in our blood, in the pH in our stomachs. We have a few different pHs in our body. Mostly everything is around pH 7.4. All right, so let me actually read the definition of isoelectric point now. The pH at which the net positive and negative charges are evenly balanced to yield an electrically neutral molecule. This is known as the isoelectric point. Let me read this again. The pH at which the net positive and net negative charges are evenly balanced, meaning maybe if you got a plus and a minus, you just have one each. If you got a +1 and a -1, those cancel out, right? So those technically we say they balance each other to 0. It yields an electrically neutral molecule overall. We are not saying the amino acid has no charges. We're just saying overall the positives and negatives balance to 0. So we say overall the molecule is a net neutral. Does that make sense? And that is the Zwitter ion. Okay, so if I scroll back up for just a second, if the amine group has a plus charge over here and the carboxylate has a negative, and we just have one each, that is the Zwitter ion. That is when the molecule is at its isoelectric point, neutral overall. And so I'm going to scroll back up to the amino acids tables because there were numbers associated with each amino acid. I didn't talk about those numbers because I was waiting to define this. So let me go back to the amino acid tables. All of those numbers. So for example, did anybody notice these right here? 3.2, 3.0. Why are there numbers associated with every amino acid? These are the isoelectric points, okay, for every amino acid. This is when overall the amino acid is neutral. Now what you're seeing, the way it's kind of confusing on this table. So for example, look at arginine right here, arginine. Right now, the way you see arginine, what is the overall net charge of arginine? If you add up all the pluses and the minuses that you see within this amino acid.
Is it +1 or?
It is, Maggie. You got a +1 here and a +1 here and a -1 here. So overall, that is a +1. Okay, overall. So this is not how the molecule looks at its isoelectric point at pH 10.8. But what you're seeing on this table, it's not showing you the form of the molecule at pH 10.4. It's showing you the form of the molecule. I think I wrote it up here. Where did I write it? It's showing you the form of the molecule at pH 7.4, what it would look like in the body, okay? So in the body, arginine moves around, floats around, is carried around as a +1. But it is technically, if you raise the pH to 10.4, it can become electrically neutral. That's the isoelectric point, okay? This stuff is where things can get really hard to understand with amino acids and kind of confusing. I just want you to know what an isoelectric point is. I'm not going to really drill you on isoelectric points. You don't necessarily have to memorize the isoelectric point for every amino acid. I do want you to know the side chain, and I do want you to know the 3-letter and the 1-letter abbreviations. But you don't have to memorize the isoelectric points. I just want you to know that amino acids can look different charge-wise depending on what pH they're in and what kind of environment they're in. Okay. Practice with this. Amino acids. Actually, I'm going to skip over this because I'm looking at the time. We still have a lot to get to. I'm going to skip over that. So I'm probably not going to ask you anything exactly like this on the exam. I might ask you define what the isoelectric point is. Okay. Or if I give you an amino acid and I wrote in the certain charges of it, I would ask you, is this the Zwitter ion form? Is it at its isoelectric point or is it not? I might ask you something like that, okay? If overall the net charge is not 0, then it's not at its isoelectric point. But if overall the net charge is 0, it is at its isoelectric point, okay? All right, we got to get, so that's just, we've just been talking about amino acids. Now we got to get to, we're going to start to zoom out, okay? And talk about what happens when we put amino acids together. So just like we looked at with monosaccharides, you can link up monosaccharides to get long chains, polysaccharides. You can do that with amino acids, and we call these peptides. So you can link up amino acids to get peptides. So a peptide bond, and it's an amide bond that links 2 amino acids together. Notice the word amide here. You know what that functional group is. So here's what this looks like. It's an amide bond that links 2 amino acids together. So let's look at an example. We have valine and cysteine. The side chains are highlighted in green, or whatever that grayish color is. And in blue, you see the amine group and the carboxylate group of each amino acid, okay? So these 2 in the middle are the ones that I want to highlight. Here's the carboxylate group, and here's a valine. Here's the amine group of cysteine. So this is technically a dehydration reaction. We're going to lose a molecule of water. I'm going to write that. This is a dehydration. Hopefully that will help you remember how this works. We are going to lose this OH and this H right here. Those are going to leave those amino acids and make water. That's why we're calling it dehydration. We're losing water. And they're going to link up. Okay, so everything is just going to scooch together in the absence of the OH and the H, and we are going to get a peptide bond. And I'm going to highlight this right. So now these 2 groups are joined, and this is an amide. Okay, because you got the carboxyl group of 1 and the nitrogen right next door bonded to it. That's an amide bond. That is a peptide bond. Okay, so just the way with chapter 20, I wanted you to be able to identify a glycosidic bond with monosaccharides. I want you to be able to identify a peptide bond with amino acids. Okay. These figures are showing you the ball and stick model down here, but that all looks like a jumbled mess to me. So that helps you great. It's pointing out the peptide bond, but I like looking at it above how we just did. Okay, and then here's our molecule of water that we lost from the 2. So any 2 amino acids, all of them have an amine group and a carboxyl group. All of them can be joined up. And we could switch these, right? We could put cysteine first and valine second, and you could still get an amide bond, right? So that allows the possibility of wildly different peptide chains. And this is where you get really cool different proteins, different functions. So long polymers of chains of peptides joined together via peptide bonds are known as long polymers of as not polysaccharides, polypeptides. Almost blanked. Okay, so proteins, when we start linking these polypeptides together, proteins consist of one or more linked chains of polypeptides containing hundreds of amino acids that can get really, really long. All right. The sequence, by the way, really does matter. So sometimes, and I think I have it on a slide down below, but sometimes if the sequence is messed up by even one amino acid, that's where, like, mutations is what we call those. That's where you can get different disease, like protein-related diseases. Okay, I'm going to talk about sickle cell anemia in a little bit, because that is literally the difference of a mutation of one amino acid. Okay, so that's why we're saying the sequence really matters. If you mess up the sequence, sometimes in even one place, you can get disease states. Okay. Peptides and proteins, I mentioned this earlier, but we always put the amino terminal. Need to write smaller. Amino terminal on the left, and we say the carboxyl terminal on the right. Okay, that's how we orient them. The amino terminal on the left, carboxyl terminal on the right. Okay, so we're going to do some practice with writing out peptides. Okay, so valine, I'm going to zoom in on this top practice problem. Valine is an amino acid with a non-polar side chain, and serine is an amino acid with a polar side chain. Draw the two dipeptides that can be formed with these two amino acids. Identify the peptide bond. All right, so we're going to draw these out, but here's the two you can get. You could get valine, serine, or you could get serine and then valine, right? These are the two dipeptides. So that's what it means, but we're going to draw these out with their side chains. So the side chain for valine, if you have the alpha carbon here, the side chain, remember I said it was like an upside down V. That's how I like to draw it. So there's the side chain for valine. The side chain for serine, anybody want to go look at the table? Double check me. Ch18, I believe.
Yeah, that's right.
Yep, there it is. Okay, so let's draw this out. We need to draw the amide bonds, the peptide bonds. So let's draw this top one first, valine, then serine. So you got to start out, I like to orient myself by drawing my alpha carbon and my hydrogen and my side chain. So this is valine. So draw this on your paper with me. Now I need to draw the amine group. I'm going to draw in its plus form. If you draw as NH2 right now, that's okay. I haven't even stated what kind of pH we're at. So you could draw it as either NH2 or NH3+. Either one's okay right now. Actually, I'm going to draw in its neutral form. It might be more confusing if I don't. Okay, and then we're going to draw the carboxyl group. Okay, but I'm going to stop when I get here. I'm not actually going to draw the other oxygen with the negative charge, because when we join these up, we lose that oxygen. We lose the OH group, or if it's just in the anion form, we lose the negatively charged oxygen. And this carbon in a dipeptide is bonded right to the next nitrogen in the other amino acid. Okay, this should look exactly like what you see up here, even though we have valine and cysteine. Now we're just going to draw serine instead of cysteine. So we got one hydrogen on this nitrogen. Now we got the alpha carbon of serine, and we need to attach the one hydrogen that has, and now we need to do the side chain. So the side chain of serine we said was a CH2OH. So I'm just going to literally copy that down right here, CH2OH. So this one was valine, this one is serine. And then we need to finish serine out. What are we missing from serine? How do we finish this?
carbon needs an extra. Oh, does it need the double bonded up?
Needs the carbonyl group. We need the carboxylate portion or the carboxyl terminal. So we got the amino terminal, we got the carboxyl terminal on the right, we got valine, then we got serine. We've drawn the side chains of both. And right here, if I had you highlight, this is the peptide bond. This is an amide group overall, so that's why we sometimes call it an amide bond, peptide bond. You'll hear people use both words. Linking the two amino acids into a dipeptide. For time's sake, I'm not going to reverse it, but you literally could draw serine first and then link it to valine, okay? Same kind of amide bond. Okay, what does the next practice problem say? Tripeptides are composed of 3 amino acids linked by peptide bonds. Given a set of amino acids, you can make several different tripeptide bonds just by putting the amino acids in different orders. Okay, this one's, I think, a little easier. We can do that. Use the 3-letter shorthand notation to name all the tripeptides that can be made from serine, tyrosine, and glycine. Each amino acid should only be used once. So we need the 3-letter shorthand notation for each of these, okay? So anybody know the three-letter shorthand notation for serine, tyrosine, or glycine, and glycine? So serine we actually used right here. So it's just SER. What is tyrosine?
TYR.
TYR, thank you, Claire. And anybody know what glycine is? Always a good guess is the first 3 letters, but it's not always the first 3 letters. So is it GLY? I saw someone nod. GLY. So now we're just going to put these in different orders. So the different kinds of tripeptides we could have would be literally the order we just wrote: serine, tyrosine, glycine. Can anybody think of another way we could orient these?
Tyrosine, serine, glycine.
Mm-hmm. How else could we orient these?
Glycosine, serine, glycine.
If anybody's really into like codes and like code words and like code, you should be good at this.
Can I ask you a question really quick?
Yeah, Abby.
So you know how in like the isomers, how like, so right there we have the tyrosine and then the glycine on the end. So like it's not like the isomers where if you flip it, it's the same. Does that make sense?
This is not, because, yeah, so this one is not like if you flip the isomer, it's the same thing. So if I write glycine, then TYR, then serine, these two are truly unique from each other.
Okay.
You have the same amino acids, but okay, isomers, Abby, are kind of really only, we don't necessarily discuss isomers within peptides.
Okay.
Yeah.
I was just curious. Okay, thank you.
No, that's a great question though. But that is really only looking at the connectivity of atoms. Here we are looking at the connectivity of amino acids. But I'm not going to ask you, what's the isomer peptide of serine, tyrosine, glycine? No, I won't do that. Okay, so Abby wrote us another one. So we're up to 4 now. Let me see if I can get any other ones. We could have serine, then glycine, then tyrosine. I keep writing tri, it's TYR. Then we could get, no, we already got that one. Is this all we can get? Oh, we could do tyrosine, then glycine, then serine, right? We haven't done that yet. So we got 6. That might be all. I don't know. If anybody thinks of another one, you could write it down. But you get what we're saying. You can orient these all different ways. B says draw the complete structure of the tripeptides that have glycine as the N-terminal amino acid. So that would be this one, glycine's first. So it's on the left, so that would be the N-terminal one. And this one right here. I'll draw out one of these, but for time's sake, we're not gonna do both. So I'll just do the first one, glycine, serine, tyrosine. So if we draw this out, I'm gonna draw glycine first. Here's the alpha carbon. And we got the amine group. Glycine has 2 hydrogens attached to its alpha carbon. It's the one amino acid that's not chiral. Then we have the carbonyl carbon, and now we have, it's gonna be linked up to the next amino acid. So serine. Okay, there's the amide bond. We got a hydrogen on that nitrogen. Draw the alpha carbon now of serine. So this right here is glycine. Now we're drawing serine, should have a hydrogen on its alpha carbon, and the side chain is CH2OH. And it has a carbonyl carbon bonded to the alpha carbon, and then we're done with serine. Now we're gonna bond that lastly to tyrosine. So we have 2 peptide bonds in this tripeptide. We have 2 amide bonds. So here's the second one. I'm gonna highlight these. There's the first amide bond linking glycine and serine. Here's the second amide bond linking serine and now tyrosine. There's the alpha carbon for tyrosine. Anybody remember what the side chain of tyrosine looks like?
It's a CH2 connected to a phenol, I think.
CH2 and phenol we know is aromatic with an OH. That's great. And then we got to finish tyrosine out. So we got to add the last carbonyl carbon double bonded to the oxygen there. Okay. And this was tyrosine. So that is a tripeptide. So are you guys getting the hang a little bit of how to link these peptides together? How to orient the amino terminal? So I'm gonna write in blue. This would be the amino, or people might say the N terminal. This would be the carboxy, or people might say the C terminal. So we're calling glycine the N-terminal amino acid. And here we're calling serine, see it said N-terminal amino acid in the problem. Because it's the first one. It's the one furthest left. The carboxy terminal amino acid is the one furthest right. Okay. Be able to draw peptides on the exam, okay? I want you to practice this. So if you need to draw out more, there's more practice on this. You could draw this other one. We didn't draw that. You could draw all of these for crying out loud if you want more practice with drawing out peptides. Identify next problem. Identify the amino acid in the following dipeptide and tripeptide. Write the abbreviated forms of the peptide names. It doesn't say 3-letter or single letter. So why don't we just do the 3-letter? We'll do the 3-letter abbreviations, and then copy the dipeptides, draw a box around. We're not going to copy them. They're already drawn for us, but we will do this part. Draw a box around the peptide bonds. Use an arrow to identify the alpha carbon atoms. Draw a circle around the R groups and indicate if the R groups are neutral, polar, acidic, or basic. What a great problem. There's a lot going on here. So which amino acids, first off, do we have in our dipeptide right here? Can anybody recognize these from the tables above?
Is
So-
the first did you call it N-terminal? Is that the valine one?
So this N-terminal is not valine. Valine, I'll highlight, has 3 carbons, carbon, carbon, carbon in its side chain. This one I see has 4. 1, 2, 3, 4.
Oh,
So it has 1 more carbon, so it can't be valine, but that it does look a lot like valine. Can anybody identify this one? Flip back up and look at those tables. So this is completely hydrocarbon side chain. So this is a non-polar amino acid. So you're going to want to look at the table that says non-polar or neutral side chains. Somebody.
Is it leucine?
It is leucine. I always have to really double check if it's leucine or isoleucine, but this one is leucine. Okay, so I'll write it above. It's well, I don't have a lot of space. Whoops. LEU, leucine. What is our next amino acid? So you need to just consider this part, okay? Or you might see it Ch2C, like you might see it like this on the table. So it's going to be down with the acidic amino acids. It's got a charged side chain.
Would it be aspartate? I don't know how to say it.
Aspartate, aspartic acid,
Aspartate, yeah.
So ASP there. So these are the 2 amino acids. Oh. What do y'all think is easier, looking at the amino acid and identifying it, or given the name, drawing it out? No opinions?
I like looking at it.
Looking at it and naming it?
Easier than drawing the whole thing out.
Okay. All right, so we did the first part. Now it says draw a box around the peptide bonds, if there's more than one. So you should look in the smack dab middle, find the amide bond. Right here. That is the peptide bond. Specifically this bond between the carbon and the nitrogen is what is linking the 2 amino acids, right? But we include the carbonyl carbon for reasons I haven't got to yet. Okay, mainly because there's resonance and the electrons can move back and forth, but don't worry about that. Okay, we circled the peptide bond. Now identify the alpha carbons. So the carbon that the side chain is bonded to, that's the alpha carbon. And that carbon should always have 1 hydrogen bonded to it. So really you just look for the carbon that has 1 hydrogen bonded to it. It sits between the carbonyl carbon and the amino group of each amino acid. So try to draw your arrow before I do. So you should have drawn an arrow right here. That's the alpha carbon for leucine. And right here, that's the alpha carbon for aspartic acid or aspartate. Now draw a circle around the R groups and indicate if the R groups are neutral, polar, or acidic. So this should be pretty easy. This is a side chain. This is a side chain. And what do y'all think about leucine? Is the side chain neutral, polar, acidic, or basic?
Is it neutral?
Completely neutral, okay? If it's just carbons and hydrogens, non-polar, not gonna have a charge, totally neutral. What do y'all think about aspartic acid? Neutral, polar, acidic, or basic?
Is it polar?
It is, but more specifically, it has given away the hydrogen. Okay. Okay, so this did used to look like this, but it actually gave away this hydrogen. And things that give away hydrogens are technically acidic. So if we're being the most accurate we can be, instead of just saying that it's polar, we can say this is acidic. So anything, aspartate and glutamate are notoriously the two amino acids that are acidic. They are always giving away that hydrogen ion and sitting in their aspartate or glutamate forms, the anion forms. Okay. All right. For good practice, you should try B on your own later. Do the same things we just did, but now you have a tripeptide instead of a dipeptide. All right, I've got to get to protein structure. I can't believe I haven't even got here yet. Okay, what slide are we on?
I think mine says 13.
And mine says, yeah, 13, 14 right there. Okay. All right, we're gonna do primary protein structure and see if we can even cover secondary protein structure. I don't want to rush too fast, and then we'll cover the rest after our break, okay? So we've looked at amino acids, we've identified the different portions of amino acids, amino terminal, carboxy group, side chain, alpha carbon, how they're different, how they're the same. Now we're going to start looking at when we've already done this. We looked at when you link amino acids together to get peptides. And now we're going to start to see that there are 4 layers, okay? 4 layers overall to protein structure. The very, very, very first basic foundational layer of protein structure is what we call primary protein structure. And that is nothing more than the sequence of amino acids in the peptide chain, okay? So primary structure is the sequence in which the amino acids are linked by peptide bonds in a protein. So you might hear me say the protein sequence or the peptide sequence, the primary structure, okay? The primary structure, the first level of protein structure has everything to do with what sequence of amino acids make up the protein, okay? So I mentioned this a second ago, but I'm going to explain now. The electrons in the carbonyl carbon are delocalized or shared. I did not spell that right. Shared with the adjacent CN. So in a peptide bond, so right here, let me zoom in. These electrons in that bond can jump over right here and they move back and forth, okay? Now we don't draw it with the electrons as a double bond between the carbon and the nitrogen. You usually just see the double bond to oxygen, but they're delocalized and it makes these peptide bonds really strong, okay? Resonance always increases the stability of biological molecules, okay? So this, it's also known as, okay, this statement here that we just filled in this bullet point is known as resonance. If I had to summarize it in one word, we'd say peptide bonds have resonance. And this adds stability to the peptide bond. For time's sake, this is not really all that important. I thought it was when I made this slide, but it's really not. So ignore this figure. I'm not going to ask you anything about the planar units, blah, blah, blah. All right, this is kind of an overall summary picture. The one thing I want to point out is I really like this phrase, the protein backbone. This is very common language. Protein backbone, primary sequence, what's the peptide sequence, what's the primary structure? Those are all interchangeable words. So the primary structure is the amino acids, the order of them that make up the protein backbone, okay? So the protein backbone is literally just the straight chain of peptide bonds, imide bonds, okay? So it goes carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen. That's the peptide backbone, okay? The peptide bonds are what you see boxed in red. And then the side chains, notice they extend out. They extend out to the side, and that is what is going to affect the function and how the protein folds, okay? because those side chains, you could have 20 different side chains. Some are really big and charged, some are really small and charged, some are completely neutral and big or small.
It's always to the side.
Always sticks out to the side. Okay, the side chain will never be part of the protein backbone. Okay, I want to hit on this. I mentioned sickle cell anemia earlier. I want to come back to this because primary structure is a huge deal. I mentioned that mutations in primary structure can cause protein-related diseases, a sickle cell anemia being one of those. Have y'all heard of sickle cell anemia before? It's usually discussed in like high school biology. Literally, the mutation of 1-glutamate to valine, that is the mutation, causes sickle cell anemia. So it really hinders your red blood cells. It affects the shape of hemoglobin in the red blood cells, and your red blood cells look fibrous and not round and cushiony. And people with sickle cell anemia, their hemoglobin and their red blood cells cannot transport oxygen as efficiently as normal blood cells, okay? But we do have a fix for this. There's a whole page in chapter 18 of the textbook that discusses how they like therapy for sickle cell anemia. It's really cool. You should check it out. I'm not going to drill you on that on the exam, but I do want you to know this as an example of the importance of primary structure, okay? And what can happen if things go wrong. There's so many more, and we're still learning. We're still learning what mutations can correlate to genetic or inherited diseases. But that's just a good example of one that's pretty well known. It's a glutamate to valine mutation. All right, so that's primary protein structure. Secondary protein structure, okay? So at the secondary level, we say secondary structure is regular and repeating structural patterns, okay? Specifically, I'm gonna highlight these, alpha helix and beta sheet. These are the hallmarks of protein secondary structure, okay? An alpha helix or a beta sheet. How do we get alpha helices and beta sheets? Well, they are created by hydrogen bonding. Hydrogen bonding is the culprit for why we get helices or these pleated sheets in protein structure, okay? And it's hydrogen bonding between, notice this is bold, the backbone atoms, not the side chains, the backbone atoms. Okay, I'm gonna illustrate this or show you this figure in a second. Okay, hydrogen bonding between the backbone atoms in neighboring segments of protein chains, or I don't want this to be misleading, it sounds misleading on the page, or it can happen within a single protein chain, just 2 different amino acids, okay? The chain could fold back on itself and you could get hydrogen bonding between backbone atoms of different amino acids. Well, somebody have something to say? Cameron, I'm sorry, you've been joining us this whole time. I'm just seeing your comments in the chat. Okay. These are explaining how the hydrogen bonding happens. Okay, so it connects amino acids from various peptides together or different amino acids within the same peptide. And what just happened? So it holds the chain in a specific spatial arrangement. That's where you get the helices or the beta sheets. So what this is the lone pair on the carbonyl oxygen. All right. So for example, you don't see the lone pairs drawn in, but you see these lone pairs on oxygen. Okay. Those lone pairs are the acceptors. We know what makes a hydrogen bond. Those are the acceptors. And that creates a hydrogen bond with the positively polarized hydrogen atom from another amino acid. Specifically, the only hydrogen that it could hydrogen bond with is a hydrogen bonded to an amine group. Why could it not form a hydrogen bond with this hydrogen? Anybody?
Is it because that hydrogen isn't, like it's bonded to a carbon, so it's non-polar?
Right. This hydrogen is bonded to a carbon, not a nitrogen. So this one is not positively polarized. But this hydrogen is positively polarized because it's bonded to a nitrogen. So you can only get hydrogen bonds when we're talking about backbone atoms between the carbonyl oxygen, the oxygen bonded to the carbonyl carbon, double bonded, and then the hydrogen that's bonded to the amine group of another amino acid. Okay. Okay. All right, that is secondary structure. I'm gonna show you a little bit more about alpha helices and beta sheets though. So what this looks like, alpha helices, this is a single protein chain, but it's just coiled, it's just spiral. I mean, y'all know what a helix is. And they always rotate right-handed, okay? So you don't have to, I'm not gonna ask you that, I just want you to know they always twist a certain way. Okay, it's not random, it's always twisted a certain way. Clockwise twist. So the way this is held together, the way the alpha helix is held together is there is a hydrogen bond that connects every roughly fourth amino acid. And then you have the R groups. This is the side chain, extends out to the side. So the way this would look, if you're looking from a side view of a long helix, it might look like something like this. Okay, you have the carboxy terminal and the N terminal down here. If we kind of removed and look at the bare bone structure, this is showing you right here that you would get hydrogen bonds between, it's technically every 3.6 amino acids, but they'll say every 4. They just round up. This is showing you, so the dotted lines here that you see, these are all the hydrogen bonds. Actually, probably if I highlighted those, these are all the hydrogen bonds holding this thing together. It tells you, color codes all the different spheres here that you see, black are the carbon atoms, the green are the side chains, and look, you see those sticking out on the side, right? So those are not in the backbone, those are sticking out on the side. I really like this view over here, figure B, because it shows you all the green around the outside. Okay, so that's an alpha helix. Okay, a beta sheet. A beta sheets link adjacent protein chains. So you're not going to get hydrogen bonding within the same protein chain as you do with an alpha helix. That's one way alpha helices and beta sheets are different. Alpha helices, the hydrogen bonding is occurring between amino acids of the same chain. Beta sheet, the hydrogen bonding is occurring between amino acids of different protein chains. So it's I thought I was highlighting. Adjacent protein chains held together by hydrogen bonds along the backbone, forming a flat sheet-like structure. There are these slight bends. It's not the important part at the alpha carbon, so that's why we call them pleated beta sheets. Gives them kind of a pleated contour. But you can see this. So, for example, this is one chain. This is a neighboring chain. Okay. So there's actually, if I keep circling, there's 4 chains here. There's 4 chains kind of all side by side. The amino terminal of one chain is lined up with the carboxyl terminal of the next chain. So they're kind of flipped 180 every other chain. So if we're looking at this, here's the hydrogen bonds. If we have, I forget what's blue. Blue is oxygen, nitrogen, and red is oxygen. Okay. So this would be a nitrogen atom, and that would be the positively polarized hydrogen. And then here's the alpha carbon, and this would be the double bonded oxygen. Okay, so you see the little red dotted lines, those are the hydrogen bonds that are connecting. You got several connecting the different chains. Now remember, a hydrogen bond in and of itself, one individual hydrogen bond, is not very strong. But what makes them significant is like there's tons of hydrogen bonds that can occur within protein structure at both the secondary, we'll see the tertiary, and even the quaternary levels. Oh, last thing, I really like this figure too, because you can stack beta sheets and you could see these side chains are sticking up and down above and below the beta sheet, and these can interact with each other. So you could actually stack beta sheets on top of one another, and you can get really unique protein structures that way. Okay. So on the exam, if I ask you how are alpha helices and beta sheets the same or similar, how are they different? Could you talk me through that? They both are formed by hydrogen bonds between the backbone atoms, but they're different. I mean, obviously their shapes are wildly different. One's a coil, one's a flat sheet, One is hydrogen bonds within the same protein chain. One is hydrogen bonds between two different protein chains, links protein chains together. Okay, well, we are at time, so I'm gonna pause here. We still have tertiary and quaternary structure to go, but I think we all need a break. Let's pick back up at 11 o'clock. I need some time to take pictures of all y'all's exams, and I'm gonna email those to you guys. When we come back on our break